So I'm going to talk about protein function and some aspects of proteins. In many ways this is meant more for the newcomers, but there are aspects of protein function that I introduced for the ones who have been in last year's lecture. Now, from now on, Jonas is going to record the lecture. So last week we had the sick week. This week we talk about the introduction. Most of the slides, or some of the slides today, come from Jan Ayofran, who is now a professor at Balagne University, Marco Punta, who is now at PFAM, or just is moving to Paris from PFAM, and Kazimir Wrzeszczynski, who is leading a group in the New York Genome Institute. What do we know in molecular biology? So what's common between all these life forms? Don't say life. It's true.
So dogma?
Yeah, you two ahead. Let's. Well, central dogma is true. These guys don't understand what you mean, but let's begin with the thing that you would say describes all ways of life. If you were to define life, I'll get back to what David meant, but if you were to define life, how do you believe this would be defined?
I would say water. Usually I have this relation.
That water is good, but the trouble is we believe that you absolutely need water for the type of life forms we have, because we explain the way life occurred on Earth through water. But we're not sure it has to be water. The type of life we have, we believe really needs water to evolve. But water itself is not life. So what is life? What's the common ingredient to life? Carbon. You're really looking at it from a very chemistry perspective. That's true. You absolutely, for the life that our life is absolutely based on carbon. We need oxygen, we need hydrogen, we need water. You're totally right. But this is still the chemical level. And I'll get back to that at some point. The amount of chemical atoms that we actually have in our body is fairly limited. The amount of ingredients, so to speak. But let's go to another level, to the larger system level. Leonardo da Vinci had this idea that life is defined as something that fits into shapes, right? In some way you may argue this is like that idea that you're going for, which is chemistry. So there are some basic elements. What I'm going for is more the molecular aspects. So what's common to life in some way clearly is DNA or RNA. And that is exactly what David said. The code, I'll get back to that in a minute, as I said. What is also common to life is a particular way of living. So it is the thing that is under evolutionary pressure or under evolutionary selection. That's the thing that replicates, for instance. So DNA alone is not enough. We need the replication mechanism. This is, for instance, one reason why RNA viruses are typically not considered a life form, an organism. It's a life form, but it's not an organism. It cannot replicate on its own. It leads to enslave another cell. There's a cell cycle. There's metabolism, the way it takes up energy. So life is essentially defined by a couple of features. Some of them, DNA, would be an ingredient, like in the chemistry sense. And some of them is really a procedure. It's the way it happens, the way it works. And that really is the view of biology. Life begins. We believe that actually life began with RNA. RNA consists of 4 different nucleotides, has enzymatic activity, can live, so to speak, on its own, form structures, can exist on its own, but there are certain things RNA cannot do on its own, and some people argue, in fact, RNA enslaved proteins to help its doing some enzymatic functions. In terms of a different perspective from watching it from today, could we imagine to just have life as we are, RNA only based? Maybe, but very unlikely because RNA is not very stable. So one, yeah?
Explain what do you mean when you say that RNA enslaved the proteins?
So enslave the proteins. This is a, yes. What I mean by that, those are, of course, this talk is on YouTube. Those are the kinds of moments you pick out of my talk, and they're absolutely great for the public, right? No, so what I mean is the following. RNA cannot do certain enzymatic functions. It needs to look for something else. RNA, again, in my definition of life, it is something that self-replicates. RNA is self-replicating, but we still don't quite have this idea of an organism. A single molecule is not quite what we would call a living being, right? But let's just assume we call that the first stage of life. Not quite life yet, but that's how it sort of begins. Now, there are certain enzymatic functions it cannot do, right? It needs help. And this help cannot be given from chemistry. So it cannot just get out some atoms, and these atoms together with the RNA do this thing. And it needs larger things. And these larger things it finds are proteins, are rudimentary proteins, constructs of amino acids that the RNA uses to accomplish certain things. In that view, I meant the word enslaved, because in that view, life begins with RNA, and the dominant pre-life form is RNA, an RNA world, and proteins help the RNA. And I also use that, yeah?
But, I mean, there's this protein ribosome, I think it's called,
yes.
who uses the RNA to produce new proteins. But somehow someone has to produce the ribosome, right?
Oh yes, but that is long before that. So the ribosome is, you could call a particle, an aggregation of proteins in our body that build up the proteins. And it's not in that sense, you may call it the protein, the ribosome, but actually we're talking about a construct that has 200 proteins and a lot of RNAs in it. So on the world of a single protein, this is like, just imagine you are a protein, and then the 200 people class is the ribosome. And the 200 class room is together performing a function. That, in fact, is the way proteins are expressed, and that is clearly not what we have here. What we have here is origins of very short stretches of amino acids. And in fact, since I spend more time on it than I usually do, there in the Max Planck Institute in Tübingen, Andrei Lupas is one of those people who argues this way, and he argues we can therefore, in all the proteins that we see today, we can still see that reality because they have high preference for binding RNA and DNA, and we can still sort of see these early motifs that those days the RNA essentially found to do its enzymatic functions. And he's trying to catalog and experimentally verify that statement. That's sort of his life work. It's an extremely interesting work where he looks for protein by protein and sees, does that one region in that protein fit to that idea? So it's really the other direction of ribosome, a tiny fraction of a protein. And we categorize those. Again, RNA is not stable. RNA, essentially that is the great advantage for us of RNA viruses, that they don't really even live in the air. The air is full of enzymes that even eat the virus up. So RNA viruses are very volatile. They are very easy to destroy. So DNA is a brick, if you want, or building material that is much more long-lasting. So in that world now, the information contained in the RNA is translated into the DNA, and we have a stable universe. That is the historical background, and we get back to what you said. Oh, here's the idea. Andrei Lupas actually is on that slide here, where he really looks at that could be the origin of the region that binds RNA, and these are all the proteins that came from it over time. So how do we proceed to the next level? So now we have the level, I call it an ocean, or somehow you have RNA in the ocean, you have your water, you have that sort of swimming around. But what else do we need? in order to really have the concept of an organism.
Proteins?
That's true, but something in a world where we would in fact still have enslaved proteins, or there's still something very, very important in some sense that defines you.
Membrane, some kind of...
Yes, you need a boundary. You need a distinction between you and the rest of the universe. And you could do it with a membrane. You could imagine that this would be solved with something else. Let's turn that around. So that's an illustration of a lipid bilayer here. And that in fact is a simple cell, illustration from Gutzel. Now, the question to you is, why not just put concrete around here?
Just let something pass by.
Why?
To communicate with other cells.
So communication is way overrated. Why do we want to communicate? Why can a cell not just?
Maybe not communicate, but exchange.
What? What is it that it absolutely, even a cell that is a being completely solipsist or completely unhappy on its own, what does that thing even need? It needs an exchange, and in fact it needs an exchange in two ways.
Yeah, I mean it's energy.
Yeah, exactly. It needs energy. So it needs an intake of energy. All the movement in the cell costs energy. And that has to come from outside. Well, you could imagine that it sort of has this energy stored inside and eats it up, and then it dies. But that's not very long living. The other thing is it creates things that it has to get out. Garbage. Disposal, right? It's just like a city. And those are clearly very two important things. But there's another aspect. And again, I said life is defined also as cell division. Metabolism is exactly energy in and out, or the exchange of material. But cell cycle, or the cell division, couldn't, I mean, if you, well, it would be complicated to imagine. Well, actually, you could imagine it like an eggshell. So then somebody cracks it open, but then you're no longer and then the eggshell, well, you could imagine it. Okay. So cell division is not the argument. I take that. But anyway, clearly we need something that is semi-permeable, so that where exchange can happen. So how densely packed are cells? And this image already indicates all of this is part of proteins, DNA, fibers. Cells are really extremely densely packed. So imagine a cell, for many years I imagined a cell like essentially a bag of water with things floating around in there. This image absolutely tells you that is the wrong image. A cell is much more like a densely populated city, like a rush hour subway car. That is much closer to what a real cell is like. This is a eukaryotic cell. And now we get to the central dogma. Life as we know it today, the organisms we know today, essentially all follow something that has been dubbed the central dogma of molecular biology, where DNA has the information, the code, the library, the manual, RNA is the intermediate step, and protein is the machinery of life. So this is four nucleotides, slightly different, and this is translated to 20 different amino acids. RNA, I already said that, also has a living function, has an enzymatic function, and in fact, for instance, in the ribosomes, it really has an active role, so it is not really an intermediate step only. It does more than just the intermediate step. I'm confused. I gave you these numbers, right? Yes. Most of you. It's a repetition. So this is just a set of different pictures of proteins, where here is the DNA wrapped around it. Those are in fact proteins, and that in fact would be the protein on the binding site view, if you want. Now what is a protein? One way of saying, defining that is it has a certain set of structure. It is something that, and you always see in proteins, for those of you who are new to that lecture, there are two major, what is dubbed secondary structure elements. Here in red, helices, in green, beta strands. So you imagine that the chain goes like this, comes back, another, and they bind. They're stabilized by hydrogen bond formation. And a mixture of these two. Sometimes you see sort of predominantly beta strands. Sometimes you see predominantly alpha helices here in the membrane protein. Also in other proteins. That is just one example of a membrane protein. This is a pore where, in fact, you look from, that's the lipid bilayer here. This pore, you look, sits in the membrane like this. And this, in fact, is the gating point. Cation gating is a very, very new anion shell that just was published. Those are two typical proteins, so-called cytochromes. Cytochromes are amongst the proteins that we believe are sort of the oldest proteins that we know. And here are two different versions. This is the peppermint cytochrome P450, and that's the spearmint cytochrome P450. And essentially, just looking at the first row quickly, one towards the other, you see there's a lot of similarity, and there's some difference. And some of that difference has to do with why that tastes different from that. But not everything. And the trick about predicting protein function essentially, in some sense, is to tell me which residues are unimportant for the taste difference and which residues, in fact, bring about the taste. And again, so the way we translate the code is transcription. You take the DNA, you transcribe it into RNA. There's a subproduct, the RNA, then in the ribosome is transferred into proteins, translated into proteins. The way that is done, since we have 4 nucleotides going to 20 amino acids, we need a triplet code. So 4 times 4, 16 is not enough. 64 is too much. That's why you in fact have a redundant code. So in this particular case here, some amino acids have more than one triplet they code for, and some amino acids have fewer. And some of them are in fact not even used. For instance, some of them are used to say stop translation. This is essentially the genetic code. Here's another view of the genetic code that essentially says first, second, third base and relates the amino acid to it. You see amino acids briefly out here in the outer ring. You see, in fact, the occurrence of those amino acids at different species, but you clearly see they occur at different frequencies. So the height of these bars here is different. That is what you can immediately see. Okay, so much. Just wanted to put it here so that you have it for your reference. How many are in proteins? How many proteins are in human? We discussed that, about 20,000. 20 to 25,000. For the newcomers, do you have any idea how long proteins are? So the units, again, are amino acids. When strung up in a protein, we refer to them as residues. It's still the same thing, except for an amino acid typically is with that particular one. Or amino acid is just the same thing isolated. While when they bind them, you call them residues. Biology is a lot about words. How long are proteins? Give me a guess. People who are new. What's the shortest protein? What's the longest protein? The guess has to come from somebody who has not attended the previous lecture, please. Oh yeah, no, I mean number of units, so to speak. Number of residues, number of... Yes, nanometer is a very, very good ballpark. Proteins are, so right out with that. Protein, the size of a protein is in the ballpark of several nanometers. That's exactly what it is. But how many residues would we have in a protein? Yeah, thousands is a good example. In fact, thousands is a little bit higher than the average. But it spans from the shortest thing that we still consider a real protein is 35 residues long. The longest in human is 30,000 residues long. And you immediately see that we have completely many orders of magnitude here, different things. And then to talk about a median here of around 400 is... formally that comes out. Was the distribution a while ago, a little bit over a year ago. Now the number of all known proteins is more in the ballpark of 70 million, so it has over doubled since.
Do you know what the longest is used for? So is it for the structure of the cell?
It is a structural protein. It's called titin, and it, of course, I believe I'll get back to that in a minute. It has many subunits. So it is made up of units that are strung up, and these units are the kind of things that you would call the original proteins, or would consider to be more like the original proteins, and they are typically referred to as domains. Domains you can imagine like pearls, so compact three-dimensional units, and then if you string up many of these domains, it's like a pearl chain in some sense again. And titin is in fact one that is made up of many small chains that does form fibers and has a bunch of functions. The problem studying titin is just so incredibly big that we still don't quite know what the whole thing is. So there are fibers, actin fibers, that we really see. So you saw some in the cell, and I'll show you images of actin fibers again. So you can really see them as tracks. Titin is not like that. It's huge, but it's still not completely clear. It's made up of components that have all kinds of functions, and it forms fibers, but there is more to titin than that, and that is still not quite answered. Again, there's a distribution here of length, and at this point over 70 million are known. So here's one issue that has to do with why it is so important to predict protein structure and function from sequence. And these numbers are done a little bit over a year ago. In 2009, September 2009, we had about 8 million proteins. And June 13, we're 35. This is more than double every 2 years. This is faster than computers now grow, right? But the story gets slightly more complicated. If you want to do anything about a protein, if you want to predict structure or function, you always begin, say you started with a peppermint, you always begin by comparing it to whatever you already know. As a database comparison. The database comparison, like this experiment here, in fact, the number of comparisons for the entire database here is 6 by the 10 by the 12 in 2009. Here, we have 1.2 by the 10 by the 15. That is a 20-fold increase every year. That is, in fact, the CPU you need. This is not the CPU you need. That's just the, and it's not even the disk space. The disk space is also here, because every one of these events you want to store in some way. And that, in fact, is immediately visible here. Now then there is some compression, and not everybody is, you have to store. So, but you still have over fivefold every 2 years increase ever since. And you immediately see this is something where, in fact, the computational data grows faster than the resources we have at hand. And that is one of the reasons why we have a problem and need many people in this field. Let's put it as some facts. And now the animation. So again, I said that they consist of units. Most proteins have more than 2 domains. So the same sequence always falls into the same 3D structure. Does the same protein always have the same sequence? Mostly. Side chains, so those are the 20 amino acids. The way they are built up, so that essentially is the basic skeleton of an amino acid here. And they all in principle have that. And then they differ by what they put on the CH3, on top of this CH3, and that is considered the side chain. Here in this particular case it's going to create a charged amino acid. Here's the second charged amino acid that can be through protonation charged, the histidine. Again, you don't need to remember the names of the amino acids, but it is important that you know that they have certain simple features, such as charge, and they differ by that. Here's a negative charge. Here's glutamic and aspartic acid. Then they are polarizable. Asparagine glutamate, serine, tyrosine, threonine. And then there are hydrophobic amino acids. Hydrophobic amino acids, simply they, if you want to think about it, they want to stick together. When I exclude water, yes?
Just a question about why it has to be like the same sequence falls into the same structure. I mean, it's like more like an optimization problem, isn't it? And maybe there's two solutions for that. So it could, I mean, it tries to minimize the energy by folding.
Yeah.
But why isn't it two solutions, two global minimum?
So,
or many? Why it has to be one?
Let's begin with a repetition. Let me see. Let me get through a few more slides, and then I get back to that point. What is a protein structure? Again, this is the shape here. What you see here is a ball. Essentially represents a residue, and you don't really, in this view, you just see a bunch of blue and a bunch of red. If you put sort of, you label them in a different way, in which you have a trace here, a ribbon, and then you in fact use the cylinder to illustrate a helix, you immediately see a theme coming up. Here's a different theme, and that different theme in this particular case is the alcohol dehydrogenase, is the protein that is important to eat up alcohol. Now, let's get back to what our current belief is. So first of all, the first statement. Our belief is that you take a sequence, you throw it into water or solvent, and it adopts a unique three-dimensional structure. That is, we know, not the case for many proteins, but we do know that is the case for many proteins. Whether the majority of proteins behave like that, at least the short ones, or not, we still don't know. We tend to believe the majority of relatively short proteins, they behave like that. So we believe that there is one solution, that there is one global minimum into which they fold. We believe that in principle, if we knew the environment and we knew all the forces, we could in principle simulate that, and in fact in a computer run a folding simulation that would give us the folded structure. Today we cannot do that. We can do it for fragments of proteins. We can do it for very short proteins. And the most successful we can do it is one machine called Anton standing in New York that has by far the world record at getting the longest time of simulating the folding. The folding is a process that happens in the ballpark of microseconds to minutes. Microseconds nobody can simulate. But getting close to a microsecond, that is what Anton can. And this is a specialized machine for molecular dynamics that in fact does exactly that. And it's more than an order of magnitude faster than anything else in the world. But so you could now argue that's a proof of principle, that there is one solution, because Anton finds one solution. Actually, it's an ensemble of solutions, but this ensemble of solutions does not really correspond to what you have as a bimetal switch state. So in your question, David, you had this idea, why is there not two stable state, and it's sort of bimetal-like flips between the two? So what this folding simulation shows is that there are metastable states in between. As a technical issue, you introduce a little bit of temperature and you overcome these metastable states and you end up in a global minimum. That's the solution so far. But I told you we cannot really do long proteins and we cannot really do minutes. So currently we can call that only a belief. Okay? Now let's get back to the other issue. We believe that the particular way this structure looks, so the particular minimum it has adopted, is very important for its function. If we had a binary state, you could argue two things. You could say, well, that is great, because actually this protein could do two different functions. Yes, the trouble is, when it comes to a certain place, it may just be in the wrong shape. Right? So it's a Vabon play, whether now you could imagine, okay, maybe it takes this state, if it is next to DNA, and it takes this state if it is not next to DNA. And that in fact is what we see. But this is a different energetic solution, and this is not what you mean. So this is, I sort of intrude into the environment and I change the energetics of the situation, so that doesn't work. Okay, let's just go back to that again. A protein that sometimes looks like this and sometimes like that. It sometimes does this function and sometimes this function. And there's just no way that would be beneficial. Right? It could be, we get into, well, okay, I retract that.
Depends. I mean,
Yes. I retract my statement. I can imagine ways in which it can be beneficial. But if every single protein would be like that, then I can immediately see many ways in which it would absolutely not be beneficial. So we need some reliability. We need this protein to do this thing, or some of the proteins at least, to do that enzymatic reaction. There's possibly nobody else in the enzymatic reaction, otherwise it wouldn't happen. And if it's is a crucial reaction that prevents the cell from dying, then that has to really happen. So the fact that the protein goes to one solution is something that essentially life relies upon. Still, at this point, we do not know whether all proteins do that. We do not know whether, in fact, arriving at that particular solution is a product of some additional process. For longer proteins, we know they go into, there is a pot, Groyel is one name for that. It's a big protein that looks really like a cup. Cup has a lid. Proteins that are not nicely folded go in there and come out nicely folded. And the current assumption is that this, what happens in there, is not only passive. This is also the case, but there's some active process going on. So the active process could just be to unfold and create the environment to refold inside. It could be more than that. We don't quite know that. So that would be compatible with the idea that could be still two states for many long proteins. And we need the chaperone or something like that. The Groyel is a chaperone to in fact help it to find the one that is right. But still, that is why we believe there is only one solution. And again, we cannot find it in the computer. What is protein function? Now, in this particular case of the dehydrogenase, alcohol dehydrogenase is an enzyme. That's the sequence. It's a homodimer. It functions as a dimer. Homodimer means you have, that is one protein, that's the other protein. Both of them are 95 residues long. And they just come together and form a dimer, so a pair of two, just like marriage, of the same. That's the way it functions. You need that in order to acquire its function. So it has an enzyme classification number, and I'll get back to that, that essentially defines what type of enzyme it is. And here is now a description of the function. It is an 80K is the size of the protein, kilodalton, enzyme that catalyzes the fourth step of the metabolism of fructose before glycolysis. In the fourth step, blah, 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 blah. So this is a longer description of the detailed way in which this functions, and it actually covers several aspects of it. It covers the chemistry, and it covers the process in which it's involved. So this chemistry, if you understand that sentence, if you understood that sentence, and if you had enough time to read it and understood it, you would realize it's not only about the chemistry. It's also about, in a longer pathway of action steps of chemistry that happens steps, this is one of them. So being one of them is also part of its function. Okay? Because in the cell, you could not function if one is taken out. This is crucial to get rid of alcohol, but there are other steps in there that are crucial too. This is just the sort of final step, the most, in fact, this is the one that typically is absent in most Asians, which is in fact one of the reasons why some Asians in fact have more problems with alcohol. That is that protein. But again, so it's only one step in a long line of processes. So now the enzymatic classification, enzymes are classified by the enzyme classification. The enzyme classification has 4 digits. The first digit sort of gives the big overview. For instance, it is oxyreductase. The second one essentially says what is the donor, the group of donor, and then we get into further detail. So this is the same as the sentence, but somehow put more into bullet points, put more into something that you imagine you could put into a computer. That you could sort of clearly, this is something that you could put into your computer. This number is something that you can compute with. And I will, in fact, give you a seminar in 2 weeks, I believe, from now, in which Janet Thornton has developed this method in which he really tries to bring it to a point where you can compute on the level of chemistry of enzymes. Now, this is what I meant by the pathway. KEGG is a database of the Kyoto Encyclopedia, is a aggregation of pathways. Pathways are simply things such as this ADH is one of the steps in this pathway. And there are hundreds of these pathways in our cell. And proteins come together. Some proteins are unique to some pathways. Most proteins come together in different pathways. In particular, enzymes are typically spread all over the pathways. Again, function is the chemistry this does. Function is also where it is in this pathway. And function is being part of this pathway in some sense. And that immediately makes it more complicated. Now this idea of you have different aspects is put into what was called the gene ontology. Michael Ashburner started it, and Susan Lewis. And again, so the gene ontology is the attempt to look at something like the EC classification, where we have for enzymes, and come up with numbers that would categorize proteins, would introduce an ontology. It started originally with fly, because Michael Ashburner was a fly geneticist, and he just wanted to, one by one, make sense of the genome. That's how the whole story started, in his private computer at home, or in his lab. Now, there are, in fact, you see, all of these here are things that you can say about that particular dehydrogenase. It has these enzymatic functions. It has, you can say, well, on a coarse-grained level, it has catalytic activity, cleaves some bond. It is part of this, and all of this is part of the Go ontology. Now, what is protein function? And ultimately, the answer is we don't quite know. It's not very well defined. It is all of that together. And it depends really on what your interest is. You could, on the level of the chemistry, really ask, how is this particular atom bound or cleaved? The biochemical is really how is the enzymatic activity done? On a cellular level, you would argue, well, is that part of a cell cycle? Is that part of what duplicates the cell? Is that clearly is an important aspect. On a developmental question, you would ask, is that something that happens at some early point of the cell development? Is that something that sets about other things? Is that some crucial bottleneck for the cell or the organism to become the way it is? Hairless, or there are these particular proteins that if you put them at a certain point in the frog, it has a second head or something like that. Those are developmental activators. Is it physiological or in some sense related to a disease? Is it on a genetic level dominant or recessive? So they're very different levels, and they all are part of what a protein does. And they're all, essentially what is protein function is determined by what you are interested in your research. Okay? For us, function is anything that happens to or through a protein in our group, and we try to predict it in this sort of action sense with a focus on this realm here, biochemical, atom, cell cycle, somewhere in between is where our focus sits. And that, for the course of the lecture, will essentially be sort of the operating definition. And then we will simply go one by one in examples for aspects of function that are more easily definable. And then I will simply address the question, can we predict those? The Go annotation that I showed you before is one that Tobias will talk to you about next week. So again, protein function, everything that happens to or through a protein. And our goal is to predict protein function from sequence. That's our group. Now, back again to the question, can we compute? We cannot quite do that. We cannot predict structure from sequence, and we can certainly not predict function from sequence. But the first step is always to find similar ones. The programs to do that mostly is PsyBlast. So PsyBlast, essentially what it does, it compares to proteins letter by letter. We talked about that last semester. What I'm going to talk about during this semester really is not how BLAST works. Just imagine it is a fast way to compare all against all. But what is it that we can do with it in terms of how well can we predict function? Again, back to these two issues. I mentioned that already. Function is known for 3,000 to 12,000 of the human proteins, unknown for another fraction. The reason why there's such a big spread is simply, do you want to say this protein binds DNA, and if you know it binds DNA, you know its function? Or do you really want to say it's an ADH and know precisely the biochemical reaction? And that is the reason why there is such a spread. We know much more coarse-grained for this is a particular enzyme, but we don't quite know what its role is. We don't quite know what its biochemistry is. In some cases, we even know the biochemistry, but we still don't know the cellular role. Titin is an example for that. We know a lot about the biochemistry, we know a lot about the function, but the entire cellular makeup and process we still don't know. Now, I said that in principle we believe that sequence uniquely determines structure and structure uniquely determines function. So from sequence we should in principle be able to get the structure, but there are some issues with that because it's a very, very tough problem. And I said that in the last semester, so I'm going to gloss over that. I went through all these slides and I overlooked that. Now, molecular dynamics, as David just defined it, is the attempt. I need some light, but it's good enough. It's the attempt to use first principles to take a sequence and in a computer simulate the way a protein folds, or the way a protein adopts its three-dimensional structure. Another aspect of function is that proteins are not bricks. So they don't function by just being like this. They function by being like that. They constantly move. And function is about that motion. Okay? Interaction in chemistry is always a thing in motion. And okay. Here we have some motion at least. And the other one we didn't. So here it is about pushing these 3 ions through, and that in fact is a filter or a pore, a channel, and here's seen from a different perspective. And you see these things, they are in constant motion. And in this constant motion you also see that the cleft here opens and closes up. It's a constant activity. And this constant activity is very, very relevant for the function. And capturing that, in fact, we clearly need molecular dynamics. But we cannot do that starting from a sequence. This kind of thing we can only realize the moment we have a three-dimensional structure. In this particular case, that three-dimensional structure came from an experiment. It takes a long time for the people who are new to do this, because you cannot look at a microscope and see it. You need to crystallize. In this particular case, it was crystallized. Then you put it into a synchrotron, and then, if you're lucky enough, and it was crystallized, and it was a good crystal, you get, in fact, the structure. Sometimes that takes many, many, many years. Sometimes it's much faster. This is an example for a protein that took many years to do, and this is an example for a protein that took much more than $1 million before you could see that image. So if you want, in some sense, the only objective was to paint that picture, And that picture costs you 1 million or more than 1 million in this particular case. Some proteins are cheaper. So on average, we believe today is about 100,000. But still, they are very, very expensive. Okay, so it's very complicated to do this. Now, once you are there to understand how the mobility is, how the motion is, some of that you see in the structure. And some of it you see in the structure because you see different solutions, which in some sense back to the issue of metastable state. They are not quite metastable state, but they are sort of wobbling around. And you saw that here. Maybe I can restart the animation, by the way, that is from Marco Punta. You see they wobble around here, and you can now imagine different freezing points will give you slightly different solutions. Different crystallization points, right? And slightly different solutions then would in fact lead to, sometimes it's here, sometimes it's here. So, and that in fact is a change that you see. You would see that, these points here are always the same. And you would see that there is a tiny variation in these points. So you would see the structure somewhere has more mobility than elsewhere. And that immediately teaches you something about function. But again, the simulation that you see here, from Marco De Vivo, in fact, did it, from Geneva, and Marco Punterherbt, this simulation was possible only because the three-dimensional structure was known. Take the structure, you start from the coordinates of the structure, and then you sort of introduce temperature, and you wobble around. And that is what MD works very well for. So that Marco De Vivo could do in Geneva. For that, we don't need the Anton in New York. Okay? So here's the other aspect, and that immediately is a limit of, I cannot imagine anything that a protein could do without being faithful to its three-dimensional shape, which is usually its order that makes function, but then there is also something that is called disordered regions. And they are quite important, in particular for eukaryotes. The idea is, in this particular case here, you come with a stretch of amino acids. Let's call it a protein. This is a different protein. There's a blue and the red one. And the red one here is sort of wobbling around. And the red one, the definition of disorder is here, you shine light, if you could, shine light onto this and see it, observe it. At different time points, you would see something else. Okay? While you shine light on the blue object, you always see the same thing. Again, within the limits of what I showed you before that motion. But essentially, you sort of go further away, you get a higher level of abstraction, then the blue stays the same, the red completely changes. Now the red approaches the blue, it binds, and upon binding, you see there is sort of this helical structure that is adopted. So the helical structure now is such that if you froze it, if you could do that, and it would just in an image, you move it away, now you would shine light and you would at any time point see the same thing. Now it's stable, now it's frozen in terms of its three-dimensional structure. Now it adopts a unique three-dimensional structure. And that is a functional principle, and there are in fact different reasons why this kind of disorder happens. That is one, to adopt this, to in fact fit to another structure. So it's induced fit, and you could imagine that you could induce fit yourself to different things, provided somebody else regulates how you get there. So this would work for some proteins, where in fact an alternative state would be helpful, as long as I have some additional regulation. There are other ways of using that. There are many ways in which we can use it. One way is if you have a long loopy region that flops around all the time. Usually if it would be compact, it would be like this. And you immediately see the amount of space you can sample with this one here is much, much bigger than what I can sample with this one. How could we use that? You never traveled in a very full subway? So sometimes in a very full subway, I'm sort of getting upright to just get a little bit more space for, and because this is not for long-term a quite comfortable position for me. So sometimes I stand up, so I create space around me. That's exactly what these guys do. They imagine filling material. So they essentially keep some space around them free. That's one action. And then you immediately, there's another mechanism that is very related to that. You sense what is around you. If something comes, you react. And that's another one. And simply by having this longer arm, so to speak, again, my longer arm here is the way I picture this. It's not really a longer arm. In some cases, it really is a longer arm. But it's also an analogy. So by having a longer arm, you can the space here you can sample and can sense is increasing. That's another function you have. There are different functions. We still don't quite know all of them. This order is very important. In particular, it's important in eukaryotes. So let's get back to protein function, evolution. I already said that. It's intuitive metrics. What is a metric?
It's I would say mathematical functional possibility to compare things.
Is that enough? So, but what David said. Well, so essentially, let me a functional way to compare two things. So I have two proteins, A and B, or two features. I shouldn't call them proteins here. Two features of a protein. To AF, to BF, I have a particular feature of a protein, and I put a function on it, and that is a metric. Everybody happy with that? No? Somebody else? Can anybody refine that? Certainly it's true, but it's not complete. What else do we need for a metric? Anybody? Does that help anybody? So let's talk about it in terms of distance. So if the distance between A and B is larger than the distance between A and C, or smaller, or something, that implies something about the distance of C and B. That's the triangular inequality. Okay? So anyway, a metric is something that clearly we need to be able to from the comparison of A to C and A to B, infer something. If that is 50% similar, and that is 50% similar, then the idea of a metric is clearly that this is sort of on the same, that means the same thing, right? This is the case, in fact, that is like that for e-values that compare sequences, for pairwise sequence identity. But already on the level of sequence identity, for sequence comparisons, we can't really put the triangle inequality in there, because we can compare similarity, not distance. For metric you need distance. For sequence comparisons we already don't have that. For structure, we essentially, RMSD is a root mean square deviation. Essentially is the question, when I look at two three-dimensional objects, and I look at them amino acid by amino acid, or residue by residue, point by point, then you can compile a 3D or an angstrom distance between them, right? And you can just sum up the square differences. That's one distance, which again is not complicated to make a metric out. What about function? So one way of doing that I already talked about is the enzymatic nomenclature, the EC numbers. Remember I talked about the EC1 is oxygen reductases. Here are the others, and that goes into further levels. And we end up with numbers here, 4.1.1.2, that suggest that they are like a metric, or that they at least, but you begin to see one complication. So this is not 4,112 and this is 4,111. In fact, we have a similarity constructed through a tree. So you need to go through a tree similarity. And that immediately makes things more complicated. In particular, and we get to that in a minute more strongly, because for oxyreductases or lyases, what is on this level is not necessarily the same thing. And if you really want to compare and compute, then you need on all levels to have really the same hierarchy, meaning the same thing. The way this hierarchy here is constructed is by experts who look at the reactions and say this is a new reaction, and this in fact belongs to that, that belongs to that, and then we need to open something new and we get further down, and then they at some point declare this explains everything. But for them it's not about comparing something that is in here in terms of its function to something that is in here. That was not the reason to come up with this classification. Much more complicated is the gene ontology. I talked about it, I mentioned gene ontology before, because it begins by acknowledging that function happens on many levels. It argues there's a biological process, a molecular function, and a cellular component, localization. Localization would simply be, where is my protein in a cell? Okay? Is it, for instance, in the ribosome? If it is in the ribosome, I already know something about what its large process is. It has to do with translating proteins. Molecular function would be the enzymatic activity. The biological process would in fact be the DNA metabolism or something like that. In this particular case here, there are the three levels. And you immediately see that the first thing is you can absolutely not compare these two levels. Let's just ask within one level. And within one level, there clearly is also not a hierarchy, because when you go down to the tree by 4, you're not always getting the same level of detail. That depends on which class you're in. And that makes it very complicated to compute Go. Here's an example. Again, this is one particular part of a larger reaction that happens in the membrane. It is in the oxidation process. It's in the inner membrane of mitochondria. It has this Go number. There's an electron transporter transferring electrons, and several aspects of that. Ultimately, the function comes about by this complex of a cytochrome C and oxyreductase. That here is the membrane part. That is the functional part in some sense. All of that relates to the function. And you see, it's rather is complicated. So let's look at some simple statements. Is structure conserved in sequence? So I made this point. Similar sequence usually leads to a similar structure, and that is a lot what the last lecture was about. Let's skip this example here. That can be used, this we can apply to predict structure for about half of all some aspects of structure for about half of all the known proteins. Let's look at some asymmetry. Similar sequence is not always getting to the similar function, and similar function is not always getting to the same sequence. Here we have the same sequence that in different tissues adopts different function. How could you explain that now with this? You asked me whether proteins have a bistable state, so to speak. How would I explain that with that?
It's the environment.
So we have a different situation, right? The environment, simply different tissues may simply change the environment. What it binds to may change the environment, and that leads to the different function. But there is more asymmetry, or in some sense, we can have completely different structures. These two things here, again, you guys haven't seen structure, take my word for it, these two things do look different, quite different actually, yet they perform the same function. The binding site here looks very, very similar in its local environment. All the remainder of the protein appears to just stabilize the binding site. Just make sure that what the chemistry that this binding molecule here sees is the same. So the spacing of the atoms that actually touch it is identical here and here. And what is underneath is very different. So we have cases like that, where you can reach the same sort of binding interface with very different backbones, or whatever you call that, or skeletons, or bodies underneath. The domain problem is another issue here. So assume now we had this one here, an ATP sulfurylase. or whatever. And now we find another protein that is similar to it. Okay? Then we may be inclined to say that in fact it's the same thing. But it turns out this is a synthase. It's a very different enzyme. How come? And ultimately, the answer most likely is related to the domain problem. So most likely it seems that the similarity here is not the thing that makes this a synthase, and that is not the lyase. So this is not the lyase nor the synthase region. Most likely the synthase region is somewhere here, and the lyase region is somewhere here, and this may be something totally different. That's the domain problem. So when we have annotations, most annotations are not such that they tell us where the function is coming from. So I said that for almost half of all the human proteins we have some knowledge about something, but for very, very few we know really the binding sites of the activity of the function. So this problem is an important problem. Moonlighting proteins. Any idea what that may mean? Term comes from New York. The moon is very, very big in New York. No, there's another reason here. So the idea of moonlighting, it's an English term, comes from this idea from actually Dorman in New York. So New York City is expensive to live in. For most people, just a regular job is not enough to pay the price for the apartment. So one job is not enough, you need two. So you do your regular job during the day, and at night you moonlight and become a doorman. That is the original term for that. So you put another job on top. That's the moonlighting idea. For the protein, immediately clear now. So these are proteins that have different functions. Here's a list that was compiled over 10 years, for 15 years ago, and you already had many. By now we know more. In some cases the situation is just like this. There's different types, different localizations, or different cell types, but sometimes different binding of cofactors. So you bind DNA or don't bind DNA. Sometimes it's a different oligomerization. That means I showed you the ADH and I said it has to be a homodimer, so it has to function as dimers. Now, in some proteins actually can also aggregate in other meric forms. And the other meric form, it in fact often has a different function. Complex formation simply is what other protein it binds to, and in some cases really the proteins have different binding sites. And these different binding sites, essentially one side of you does one thing and the other side of you does another thing. Whether this now is a feature that is prevalent, so are we expecting this for most proteins, or are we expecting for most proteins that they have one defined function? We absolutely do not know. So at this point, we are still in the situation where we have a list. The list is much larger than what I showed you before. We have hundreds of cases now where we have several functions, but we are nowhere near being able to answer whether that is like that normal or is an exception. So far, they are the exceptions, because most proteins we don't have the full functional characterization for. As simple as that. Okay, so today we have 20 more minutes. Today we have a short day. It is about, hi, you changed.
I do.
Yes, you're in 20 minutes. So, point is, we can, except for the particular example of protein of enzymatic activity, and I will give you a lecture on that, we cannot really compute function. And the way to make enzymes become something that for which you can compute the function is by bringing it down to the chemistry, to the action, the atom that is bound, or what is in fact happening to which atom. That's the only way where we can really put something that is related to a metric. Everything else is a far cry from metrics. still, we need comparisons. The best thing is the Go, the gene ontology. And Go is very, very poor. Tobias, next week, will explain to you how you can use gene ontology, how you can predict gene ontology by relating similarities, sequence similarities, and using some rudimentary machine learning. And then when I return, I will bring you a little bit more of some images. What are the limits of these comparative modeling kind of approaches? And as far as I remember, then we will get into protein subcellular localization prediction. So protein subcellular localization, in terms of Go, is one of the three main classes. So there's the molecular function, so how you do the chemistry in some sense. There's the biological process, so you're part of the process that aggregates a particular amino acid or digests a particular enzyme, vitamin. And the third one is where in the cell you are. That's what I'm going to talk about, the methods that predict where in the cell you are. Where in the cell you are, in some sense, again, one of the reasons why many groups got into that is because it appears to, so my background really is in biochemistry or structural biology. So for structure, I can see the position of an atom. I can compare structures, they're three-dimensional objects, it's very easy, very clear. Now, localization, subcellular localization, is something that in some sense is also clear. I don't need an expert to tell me where that thing is, I can measure it. It's a real reality. And in that sense it's appealing. The downside to it is subcellular localization is either something that is stable, like a nucleus has a membrane around it, but then there's transport in and out, but still this is something that I can really see. But in the case of a nucleus, I just have a lot of proteins in there. So as a functional descriptor, that is helpful, but so in the nucleus I typically have in the ballpark of 1,800 to 2,000 proteins, so 1/10 of all the proteins. So most of the proteins in there bind DNA. So by saying this protein is nuclear, I already have an idea it binds DNA, just in a statistical sense, and I do way better than random. So in that respect it's great. In other respects, you want really to know what that particular protein does. It's far, far away from where we want to go. Anyway, any question? Thank you.
Please.